In this video, we'll take a look at a few defense responses in plants. Some of the main threats to plants include the presence of pathogens that infect plant cells or herbivores that consume plant tissue. In order to protect themselves against these threats, plants have developed some mechanisms that respond to attack by either pathogens or herbivores. There are a variety of protective responses for the plants. Some examples include insect repellent properties of peppermint and lemons. Pitch that oozes from pines and firs contains pinene. The pyrethroids produced by chrysanthemum plants are used in commercial insecticides. Tannins sicken herbivores. Compounds like opium, caffeine, cocaine, and nicotine disrupt the nervous systems of plant-eating insects and vertebrates. How do plants sense and respond to pathogens? If a virus, bacterium, or fungus invades a plant, the plant mounts a defense called the hypersensitive response, or HR. HR causes the rapid and localized death of cells surrounding the site of infection, starving the pathogens, preventing the spread of infection to other parts of the plant. Other HR defense responses include closure of the stomata, production of toxin molecules that target pathogens, and reinforcement of the cell wall to prevent movement of pathogens from one cell to the other. Once HR is underway, a hormone produced at the infection site triggers a slower, more widespread set of events called systemic acquired resistance, or SAR. In addition to triggering the HR, interaction between two genes releases a hormone that initiates SAR. These genes are called pathogenic release genes, or PR genes. The hormone that affects the action of the PR genes has been identified as methylsalicylate, or MESA. As we can see in this image, when a pathogen infects plant leaf cells by releasing a pathogenic molecule, that molecule binds receptors on the plant cells, triggering the HR response, which in turn triggers the release of an SAR signal that travels to different parts of the plant body and stimulates transcription of PR genes that generate proteins which protect the plant from further infection. Several observations support the hypothesis that MESA is the alarm hormone that is responsible for systemic acquired resistance. These include observations that MESA levels increase dramatically after tissues are infected with a pathogen. Phloem sap leaving infected sites has elevated levels of MESA. Treatments that reduce MESA reduce or abolish SAR. And adding MESA to lower levels of tobacco plants leads to SAR in the upper untreated leaves. All of these observations indicate that MESA is produced at the site of infection and travels to other sites distant from the site of infection, inducing protective responses in these distant regions. How do plants sense and respond to herbivore attack? Many plant seeds and storage organs contain proteinase inhibitors that block the enzymes found in the mouths and stomach of animals that digest proteins. When an herbivore ingests a large dose of a proteinase inhibitor, it gets sick. Over time, herbivores learn to detect and avoid plant tissues containing high concentrations of these proteins. This led scientists to hypothesize that proteinase inhibitors might also be part of an induced defense, which begins when an herbivore consumes plant tissue. Results of several experiments support the idea that a hormone produced at a damaged site travels to undamaged tissues and induces the production of proteinase inhibitors. This hormone is called cysteamine, a wound response hormone produced by herbivore damaged cells that initiates a protective response. which is the synthesis of jasmonic acid in undamaged cells. Jasmonic acid in turn activates the production of proteinase inhibitors that make the herbivore consuming the plant sick. This image shows the mechanism of function of cysteamine. Cells that are damaged by insect consumption release the hormone cysteamine, which travels to distant parts of the plant body and binds to receptors on undamaged cell surfaces. This binding triggers a signaling cascade within the undamaged cell, leading to the release of jasmonic acid, which stimulates the expression of target genes that lead to the production of proteinase inhibitors, which taste bad and make the herbivore sick. Over time, the herbivore learns not to consume leaves from plants that produce these proteinase inhibitors. Another way in which plants protect themselves against herbivore attack is by releasing pheromones that recruit help from other organisms like the wasps. Wasps are an example of parasitoids, which are organisms that are free-living as an adult but parasitic at the larval stage. Because these organisms kill their host, parasitoid attacks limit the amount of damage that herbivores do to plants. Pheromones are chemical messengers synthesized by an individual and released into the environment that elicit a response from a different individual. For example, plants produce wasp-attractant pheromones in response to attack by caterpillars. These wasps can then lay eggs in the caterpillar's body, which act as parasites inside the caterpillar's body, growing and subsequently killing the host. This picture shows parasitic wasp larvae emerging from an infected caterpillar. It is a tragic fate, considering that all the caterpillar was trying to do was eat food for its survival. This concludes our section on defense responses in plants. After going through this information, I'd like you to be able to answer the questions listed on this slide. Thank you.